So in this lesson, that is the cell, the basic unit of life. Second and fourth activity we have related to the onion. In second activity, aim is observation of onion peel cells, whereas in fourth activity the aim is observation of nucleus in the onion peel cells. So in second activity, when we are taking the onion, we should not stain it, only the cell has to be observed, whereas in fourth activity you have to stress on the nucleus. So in order to observe the nucleus clearly, we have to use a stain, whereas in second activity we don't need any stain. So in fourth activity, to observe the nucleus, we need safranin or methylene blue stain, or even red ink is enough. So in order to save time, two activities together we are going to perform. For this we need one onion. In the second watch glass we need little water. In the third watch glass we need, for observing the nucleus, safranin. If we don't want to observe the nucleus, just without stain also we can do the second activity. That is only the outline of the cells we have to observe. So instead of safranin, methylene blue if we use, then more clearly the nucleus can be observed. And in the fourth watch glass we need the glycerin. So all the apparatus which is required, once more we will discuss. So we need one glass slide, cover slip. If you take two glass slides and two cover slips, then both the activities can be performed together. Then one onion is required, a forceps, blotting paper or tissue paper, a needle, and then for observing the nucleus, methylene blue stain or safranin stain. If both are not available, even red ink can be used. Then to provide moisture for that tiny cells, glycerin is required, and three watch glasses are required, and to observe that we need one microscope. So first an onion is taken, and in this way you have to take one of the cover of the onion, and then if you want you can fold it, or in some of the onions nicely, very easily, a transparent, almost a transparent layer can be taken out. This is the main part for which we are performing this activity. By using this transparent layer we can see the cells of onion. So first we have to place it in the first watch glass which has plain water. Then take a small piece of it and place it on the slide, and take a drop of glycerin and put it over it and cover it with a cover slip. So in second activity only this much has to be done in order to observe the onion peel cells without any stain. For fourth activity you have to place it in the slide, the watch glass which has the stain. So safranin stain, or more preferably methylene blue stain can be used. Dip it for 2 seconds and then take the onion peel cell and place it in the third watch glass which has glycerin. What is the speciality of using glycerin? This is a very soft tissue. It will easily get dehydrated. In order to avoid the dehydration, moisture has to be kept, and that will be provided with the help of this glycerin. Then put it on the slide. If you want, one drop of glycerin can be added more, and then with the help of a brush the peel has to be spread evenly, not with the needle. If we use needle to spread, then it may break. Then place the cover slip and with the help of a needle slightly slantingly drop it over the peel of onion. By doing this, the bubbles will not be formed. Extra glycerin or stain should be rubbed off by using a tissue paper or a blotting paper. Place it under the microscope and adjust it and observe. The slide will appear in this way for the fourth activity. Because we have used the safranin stain, it appears light pink in color, and slightly those dots are nothing but the nucleus, and the outer line of every cell can be clearly seen. The cells are arranged here compactly without any air spaces. So with the help of safranin, it is in this way. How are the cells arranged? They are arranged very compactly without any air spaces. They are rectangular in shape. Intermediate space is not present. Then tiny spots, which are the nucleus, are seen here. So these are the parts. When we are using methylene blue, the slide will appear in this way. So when methylene blue is used, you can perform this activity to observe the nucleus very clearly. Then when you are placing it under the microscope, clamp it with the clip, adjust the microscope and observe it. When we view it through the eyepiece, you can clearly see adjustments have to be made. Then the slide will appear in this way, light bluish in color or violet in color, with the dots which are nothing but the nucleus. So very clearly we can see the onion peel cells with nucleus exactly in this way.